Biology 1108, Presentation Number 5, Systematics. The notion of evolution includes a number of sub-disciplines within biology, and one of those is systematics. Systematics is the study of the diversity and diversification of organisms over time. Diversification obviously yields evidence of evolutionary relationships, what is sometimes referred to as phylogenesis. It indicates some degree of common ancestry and can indicate adaptations arising from the line of evolution from that common ancestry. Case in point, you take a look at the illustrations of the finches at right. Those are Darwin's finches from the Galapagos Islands. And we see that the finches share some definite characteristics, but there are some characteristics that seem to vary, in particular beak shape. And obviously these birds are evolutionarily specialized with regard to these beaks. Taking a look at the Galapagos finch diversity in the Galapagos Islands, you see that the Galapagos finches are probably all derived from some common ancestor. Darwin realized this, and he reasoned this, and DNA work on the finches actually supports this notion. Now, the primary mechanism of diversification leading to those different beak shapes in the finches, or any of the diversifications in any species, be it tree or plant or fungus or animal, is through natural selection. The process of natural selection, you'll remember, selects for certain genotypes, selects against certain genotypes, and really where the rubber hits the road is those genotypes code for phenotypes where they really determine whether fitness is attained or not. If a particular genotype results in a phenotype that improves survivability or improves reproductive success, it's going to be selected for. And if it doesn't, it will be selected against. And as a consequence, that particular adaptation could then become a standard issue in that particular species or in that particular population. We have already acknowledged that while natural selection is the primary agent of evolution, it really ain't the only game in town. So we understand there is mutation, which is the way that ultimately all genes arise. Gene flow, which is the movement of genes into and out of a population through immigration and emigration. Genetic drift, the influence of random chance events on generally populations with smaller size. And then finally, non-random mating, which is the exercise of choice in determining who you're going to mate with, which can ultimately influence the genetic trajectory of your population. And as long as we're reviewing mechanisms of evolution, let's go ahead and mention the various forms of reproductive isolation, geographic isolation, behavioral isolation, habitat isolation, resource partitioning, mechanical isolation, and gametic isolation can all lead, along with the other mechanisms mentioned before, degrees of genetic variation within populations and subpopulations, which could ultimately lead to the origin of new species, the process that we called speciation. Okay, whether you pronounce it phylogenesis or phylogenesis, this is the term that describes the evolutionary origin of biological groups of organisms. Now, a group of biological organisms share certain characteristics, share certain genes, and share an evolutionary ancestry. A biological group of organisms is called a taxon or taxa plural. So phylogenesis or phylogenesis, synonyms, sometimes you will encounter words like phylogeny or phylogenetics. Basically, we're taking a look at a group of organisms trying to come up with some kind of evolutionary history, and that's going to involve a number of inferences. These are conclusions based on fossil record, based on homologies, based on distinguishing homologous features from analogous features, and lastly, we will note that homologies can be morphological or molecular. Now, I'm going to go ahead and define what homology is first, and we'll talk more about it on the next couple of slides. Homologies are adaptations that arise from some common origin. So if I was taking a look at a tetrapod, which is nothing more than a four-limbed vertebrate. Birds are tetrapods, reptiles are tetrapods, amphibians are tetrapods, and mammals are tetrapods. Homologous structures would be like the four limbs. That is the front legs of a cat, the front legs of a horse, the arms of a human, the flippers of a whale, the wings of a bat. Those are all adaptations that are derived from some common origin, forelimbs. Now, what we could do is we should note that those homologies can be morphological, like those limbs, or they can be molecular. So the fact of the matter is certain proteins, for example, that might do certain jobs physiologically might not be exactly the same. So, for example, I might find a certain enzyme in a tropical bird that I won't find in a penguin, but I will find a similar protein in the penguin, just as slightly adapted for life in colder weather. And technically, those two proteins serving the same kind of function in a tropical bird versus a penguin are homologies. So homologies are adaptations that are derived from some common origin. And the fact of the matter is, if you can say that those adaptations are derived from a common origin, there's probably a pretty good idea that those two organisms with those adaptations have some common ancestry based on those homologous features. Okay, let's take a look at exactly what constitutes a difference between an analogous versus a homologous character, or an analogous versus a homologous adaptation. We gave the impression on the last slide that homologous characters have evolutionary value, and that in fact is the case. So let's take a look what analogous factors are first. Analogous characters or analogous adaptations are similar in form and function. They demonstrate no evidence of common origin. The common origin of characters is often evidenced in development. So let's take a look at butterflies' wings and bat wings. Butterflies and bats are not related. There's a whole slew of characteristics that prove the fact that there are no significant degrees of evolutionary relatedness between butterflies and bats. However, butterflies have wings, bats have wings, and those wings allow the butterfly or the bat to fly. So these are structures that have similar form and definite similar function, but have no evidence of common origin. As a matter of fact, if I studied the embryonic development of a bat and compared it to the embryonic development of a butterfly, those structures called wings originate completely differently in the butterfly versus the bat. So development is the key. As a matter of fact, there's an old-timey phrase that's often used, and it's not on this particular slide, but I'm going to mention it anyway. It's called ontogeny recapitulates phylogeny. Ontogeny, that's O-N-T-O-G-E-N-Y, recapitulates, that is, replays, phylogeny, which is evolution. Ontogeny, the word that means development. So development replays evolution. So if I take a look at the development of a bat's wings versus development of a butterfly's wings, that's giving me kind of a view on the origin, that is the evolutionary origin of those two groups. Analogous characters, it says here, have no phylogenetic value because they do not demonstrate evolutionary relatedness. And scientists, in many cases, have to be very, very careful. They do not get led down the primrose path, because if you make an assumption that structures are homologous and therefore have evolutionary value when they're actually analogous, you could wind up making a very significant mistake in your assessment of the evolution of that organism or that group of organisms. Okay, well, I'm looking at this slide and I see in red my ontogeny recapitulates phylogeny statement. That's good. I wasn't quite sure whether I had put it on a slide or not, but there it is. So let's talk about what are homologous characters. Homologous characters have evolutionary value. They might or might not demonstrate similarity in form and function, but they are going to demonstrate evidence of common origin. I see the forelimbs of tetrapods. I see a forelimb of a human, which is an arm. I see the forelimb of a whale, which is a flipper. I guess that's a dolphin, I suppose. I see the forelimb of a cat, and I see the forelimb of a bat, which is its wings. Now, those look quite different in some cases, and they might do completely different things, but if I looked at the evolutionary origin of a bat, a dolphin, and a human, as illustrated at the bottom of this slide, I would see that the limb buds that are forming in those embryos are ultimately going to be the origin of those appendages that I see there. So common origin of characters is often evidenced in development. Ontogeny recapitulates phylogeny is a famous statement. It was made by a guy named Ernst Haeckel back in the 1800s. So homologous characteristics have phylogenetic value because they demonstrate evolutionary relatedness. And so therefore, all of these organisms qualify as vertebrate tetrapods because they have these shared characteristics. Well, you can't have systematics without classification. So classification is an activity, and it's an activity that involves organizing species into phylogenetic groups. Each group is evolutionarily derived from some common ancestor. We'll talk a little bit more about that in a moment. But classification is what humans do. It's a practice that is uniquely human. From the time you're really, really small, you distinguish between what's big and small. You distinguish between what's round or square, what's red, blue, or green. Okay, you distinguish between different types of fruits, between different types of vegetables. You go to grocery market and you basically can distinguish between all kinds of different food products. Same sort of thing. So within the world of biology, we organize species into groups. Those groups demonstrate some degree of evolutionary relatedness because they are derived from a common ancestor. For example, we take a look at the cartilaginous fishes, which would be the sharks and the skates and the stingrays. And all those organisms have some common ancestor. If I took a look at the reptiles, and all the reptiles have some common ancestor. If I take a look at the mammals, all the mammals have some common ancestor. Now, that ancestor may have existed a long time ago, and that ancestor is going to demonstrate some characteristics that indicate some degree of similarity with the groups that we see today. So when we organize these species into phylogenetic groups, it's classification, but sometimes we also call that taxonomy. Now, taxonomy is the idea that when I do have a group, that group is defined by its characteristics. And a lot of times we put a name on that group. So all the mammals are placed into a group called the Mammalia. That's taxonomy. And all the mammals share certain characteristics, and those characteristics indicate some common ancestry. So classification, that is creating phylogenetic groups, also called taxonomy, relies on the interpretation of characteristics. And those characters or characteristics can be analogous versus homologous. They can be shared or derived, and they can be non-adaptive versus adaptive. What do you mean about shared versus derived, Dr. Canales? Well, a shared character is a character that every single species within that group has. So, for example, all mammals share warm-bloodedness, mammary glands, and hair as a body covering. Now, derived characteristics are not shared by everyone. As a matter of fact, derived characteristics are considered to be somewhat advanced. So, for example, within the mammalia, we have the placental mammals. Those are mammals that have a placenta. Not all mammals have placentas, and the placenta is considered to be a derivation or a more advanced adaptation found in the mammalia. It is considered to be a derived adaptation. Another example might be within the snakes. All snakes have scales, all snakes lack legs, but the venomous snakes have special glands that produce poison, and they have special teeth that deliver that poison. So whereas scales and a lack of limbs are shared characteristics, venom and venom delivering fangs would be considered to be derived characters. Now, finally, I've got this bullet point listed here as non-adaptive versus adaptive characteristics. And it's important to understand that not all characteristics are adaptive. A characteristic, in order to be adaptive, has to definitely have some impact on survivability of the species or the ability of that species to reproduce. In other words, an adaptive characteristic must improve fitness. But the fact of the matter is there's plenty of characteristics of organisms that don't necessarily have an impact on fitness overall. So, for example, I could take a look at mammals. And mammals, yes, have mammary glands. And I could say that mammary glands are an adaptive characteristic because they promote the survival of the young. And I could maybe even suggest that hair is an adaptive feature because it promotes thermal homeostasis in a warm-blooded organism. But then on the other hand, I could take a look at the limbs of a mammal, and there's 4 of them, but I also know that 4 limbs exist in the reptiles and 4 limbs exist in the birds, and 4 limbs actually exist in the amphibians as well, and I can't really point to 4 limbs in the mammals and say that is a feature that improves survivability and ability to reproduce. I mean, yeah, it's helpful to have 4 limbs, but I can't say that 4 limbs really is going to have an influence on the organism's ability to improve its fitness. Same thing with eyes, same thing with ears, same thing with the nose. Those are characteristics that are kind of par for the course for vertebrates. They're not necessarily adaptive. So make sure you know what analogous versus homologous characters are. Make sure you know what shared versus derived characters are, and make sure that you understand that not all characteristics are adaptive. In order to be adaptive, it has to improve fitness. There are plenty of characteristics in all organisms that are not necessarily adaptive. All right, I alluded to the fact that classification is a human enterprise, something humans do. Well, classifications, at least in the world of biology, fall into a number of different types. There are, first off, historical classifications. Historical classifications fall into 2 different categories. There are artificial classifications that we're going to note on this particular slide, and there are natural classifications that we're going to find on the next slide. As far as artificial classifications, these are classifications based on similar features that do not necessarily imply relatedness. See, once upon a time, a classification might have lumped all plants with red flowers into the same group. Now I'm looking at these 3 plants here, and those plants, yes, do have red flowers, but I'm willing to bet if I was to look at the details of the structure of those flowers, if I was to look at the details of the structure of those plants, I probably would not be able to argue for their evolutionary relatedness. Just because a plant's got red flowers doesn't mean it shares a common ancestor with another red flowered plant. Well, the bottom line is an artificial classification is easy. All you have to do is remember some basic characteristic and lump it into a group of organisms that have the same characteristics. It's something prehistoric humans have been doing for a long, long time. So we're going to say that artificial classifications have a prehistoric origin, and they were driven by practical needs. I mean, obviously, if you needed to distinguish between a plant with spines and a plant that didn't have spines, or if you had to remember that an insect with a yellow and black striped abdomen could sting you and hurt you, you might be less inclined to try to eat it or grab a hold of it. It would have practical value for you. So obviously, prehistoric humans were not terribly interested in lumping organisms together in terms of their relatedness, short of being able to organize them into some practical way that would benefit the humans' quality of life. So a natural classification is really close to an artificial classification in the sense that at least you are basing your classification on similar features. But in this case, those similar features can imply relatedness, whereas red flower color wasn't going to do it for those red flowered plants. If I take a look at these 3 pictures of pit vipers, which are venomous snakes, they all have a certain head shape, they all have venom glands, they all have fangs for delivery of that venom. Those are characteristics that demonstrate some degree of relatedness, whereas they still provide me with a practical way of identifying those organisms as something I need to be aware of. So a natural classification is close to an artificial classification. Essentially, you could probably say that a natural classification is an unwitting evolutionary classification. It's still of prehistoric origin. Humans had to group organisms, both plants and animals, into groups that they could deal with that would actually provide them with some practical knowledge. Is this something I need to be careful of? Is this something I don't have to worry about? Is this something I can eat? Is this something I cannot eat? And so it's those practical needs that drove that type of classification. It just happens to be that the characteristics chosen not only were really helpful for the practical needs of the human at that time, but also indicated some degree of evolutionary relatedness. So make sure you know that historical classifications, the name implies, these are classifications that were developed early on in human history, in human prehistory, literally. And we group things artificially or we group things naturally. In either case, it provides us with some practical value for living a quality life in those times. So when we get to our third type of classification, called phylogenetic classifications, we're looking at a classification that intentionally works to demonstrate relatedness. It's a classification that has a modern origin, dates its way back to Darwin, no earlier, and is driven by phylogenesis or phylogenesis, that is the drive to create these groups of related organisms. And the relatedness is determined by these characteristics that might be shared, that might be derived, they should be homologous, and are certainly going to be representative of the organisms found in that particular group. So I'm looking at crustaceans here, and crustaceans are a group of arthropods. Arthropods are organisms with jointed shells, exoskeletons they're called, and arthropods include the crustaceans and the insects and a number of other different organisms. So if I take a look at the pictures here, I see the crustaceans. I see what looks like a crayfish, I see a crab, and I would lump shrimp in with those, but then I also see barnacles, and I see a roly-poly. Now, barnacles and roly-polies, even though they don't look like lobsters, crayfish, crabs, or shrimp, are still crustaceans. So here I have a bunch of organisms that demonstrate some degree of relatedness, some obviously, some less obviously, but they still all demonstrate features that demonstrate this degree of relatedness. As a matter of fact, crustaceans are considered to be a valid evolutionary group because of these characteristics, because they all trace back to some common ancestor. That's a phylogenetic classification. So we had used the word taxonomy as a synonym for classification. To be more specific, taxonomy is describing, identifying, naming, and categorizing those groups that we're coming up with in a process of classification. The groups are called taxa. The word taxon, T-A-X-O-N, implies a single group, and these taxa can be arranged in terms of how exclusive they are or how inclusive they are according to rank. We'll talk more about that on another slide. So these taxa can be organized into big, inclusive groups or smaller, exclusive groups. And any taxon must be identified by a name, and the name must be a Latinized name, and the name is always going to be associated with a set of characters. For example, if I take a look at the grocery store and I see apples, and I know that apples come in a number of different varieties, including Granny Smith apples, I know that Granny Smith apples are labeled Granny Smith apples, and they have certain characteristics. Well, in taxonomy, I might take a look at oak trees. Now, you and I both know that oak trees have acorns and oak trees have leaves of a certain shape. And a lot of times you just look at a tree and you know in your heart of hearts it's an oak tree. Well, within the world of taxonomy, all oaks are identified by a Latinized name, that is the name Quercus. Quercus. All oaks have the name Quercus. Now, there's lots of different types of oaks. There are white oaks, there are willow oaks, there are red oaks, there are black oaks, there are live oaks. There's all kinds of oaks, and they all have the name Quercus. They all have specific species names, but the fact of the matter is the whole group, the whole taxon, described as oaks, have a name, and that is the name Quercus, is a Latinized name, and that name is associated with a set of characteristics that lump all those oaks together. And then presumably all of those oaks have some common ancestor. And once again, that's what phylogenetic classification is all about. And we're coming up with groups that have names that have shared characteristics that define that group as an evolutionarily valid group of organisms. That's taxonomy. So we mentioned in this process of classification, we're going to take these groups, we can arrange them. We can arrange them into what are called ranks. And we mentioned that those ranks vary in terms of how exclusive the groups are or how inclusive they are. Now, here's the deal. If you take these ranks, you basically fit them into a set of cubby holes. It's almost like when you go to the store and you go to the fruit department in the grocery store and you see all the apples. And all the apples are lumped together in one area, but you also have the different varieties of apples separated from one another. Well, apples is a pretty inclusive group. Granny Smiths are a more exclusive group. Obviously, Granny Smiths and apples fall in the same basic classification. They just occupy different places. So let's take a look at this illustration on this slide. Ranks come in a number of different levels. It's what we call a hierarchy. At the top of the hierarchy, you see the word domain. Underneath the word domain, there are kingdoms, there are phyla or divisions, at least in plants, phyla in animals. There are classes, there are orders, families, genus, and species. Now, domain is the most broad, most inclusive group of all. In all of biology, there are only three domains: the eukarya, which are the eukaryotic organisms, the bacteria, which are the common bacteria, and then the archaebacteria, which are those specialized bacteria that live in inhospitable environments. But those are three very broad, very inclusive groups. If I go all the way down the bottom there, I see that fox is a particular species, and it is way, way down at the bottom there. It's a highly exclusive group. That is the species Vulpes vulpes, which is placed in the genus Vulpes, which includes foxes and fox-like organisms, and they're placed in the family Canidae, which are the dogs and related organisms, and they're placed in the order Carnivora, which are the meat-eating animals, and they're placed in the class Mammalia. And so you can see, as you move up from the bottom, these groups become more inclusive. So what amounts to be a hierarchy here is a difference in ranks, and I am going to need you to know those ranks in order, from top to bottom or bottom to top, however you want to do it. Now, there is an old-fashioned mnemonic device that is a device for remembering things, and it goes something like this: King Philip came over from German shores. It's a very, very innocent way of remembering those taxonomic ranks. Domain doesn't find a place in that mnemonic device because that mnemonic device was developed somewhere back, 1950s maybe, and domains were not recognized until the late 70s, early 80s. So that's one way of remembering it. Somebody else told me they could remember it by saying King Philip came over for great sex, or King Philip came over for group sex. However you want to remember it is fine. Okay. However, I guarantee you'll probably go with the last version because that is the easiest one to remember. Bottom line is you need to know those ranks in order. Thank you. Okay, there's nothing on this particular slide that you're going to need to remember. Nothing you need to memorize. But it's a really nice glance at this whole idea of ranks and what it means to the classification process. You will see the order Carnivora over there on the far left. The order Carnivora is a group of mammals. There are three families in a carnivora: the family Felidae, which are the cats, the family Canidae, which are the dogs, and the family Ursidae, which are the bears. And you'll see within the family Canidae, I have exploded that into, 5 different genera. The genus Canis, which are dogs and wolves, the genus Lycaon, which are the African wild dogs, the genus Vulpes, which are the true foxes, the genus Kuon, the Asian wild dog, and the genus Lycalopex, which include all other foxes. And then within the genus Vulpes, we've got a number of species. Vulpes vulpes, the red fox; Vulpes lagopus, which is the Arctic fox; Vulpes macrotis, which is the kit fox; and Vulpes camo, which is the Arctic fox. When we say that any given taxon has a set of characteristics ascribed to it, that's what a description is. Description includes morphological information, might be ecological information, might be biochemical, genetic information. It is association. That means all of those organisms within that group share these features. They are generally associated with some type species. Now, a type species is like the standard example for what that group is about. So, for example, if I take a look at the genus Pinus, and I'm taking a look at all the pine trees, and there's lots of different species of pine trees, the type pine tree might be Pinus tato, which is the loblolly pine. There are white pines, there are longleaf pines, there are Virginia pines, but the type, that is the standard example of what a pine tree might be, is the loblolly pine tree or Pinus taeta. And the same thing would be true for our true foxes, the genus Vulpes. There would be some type for that, or the same would be true for our genus Quercus, which are the oaks. And any taxon is going to have its place in a line of evolution leading from some common ancestor to the organisms that we see today. That line of evolution is called a clade. A clade is a branch of a phylogenetic tree. A clade is a path or a line of evolution leading to species we see today that are derived from some common ancestor. So describing and identifying are actually flip sides of the same coin when it comes to classification. Now remember that description is a set of characteristics that are linked to a name. Well, if you're identifying something, what you're doing is you're taking a species, an unknown, so to speak, and you're looking at the different characteristics of that species and you're fitting it to a description that matches the name you're looking for. So I can look at a white oak tree and I have no idea what I'm looking at, but I do know it's an oak. It's got acorns of a particular shape, it's got leaves of a particular shape, and it's got these characters. Now, characters come in different forms. Characters come in forms that are called character states. So, for example, I can take a look at the shape of a leaf. And leaf shape is a character with all kinds of different descriptors that can specifically describe whether the leaf's got a pointy tip or a blunt tip, or if it's got a jagged outline or a continuous outline, or it's got little spines, or maybe it's got little furry, scurfy scales on it. All of these are characteristics that can be ascribed to those characters. So characters are the general feature. Character states are the specific states of those features. But the bottom line is you're going to use both characters and character states. It doesn't matter whether it's a plant or it's an animal, but you use those features to match the description so that you know without a doubt that the organism you're looking at fits into that particular taxon. It doesn't matter whether it's Vulpes vulpes, the fox, Quercus alba, the tree, or Pinus taeta, the loblolly pine. So what's up with these Latin names, Dr. Knaus? Where did that come from? Well, the whole idea of naming is called nomenclature. And it's got big, long history. Now, history begins with the nature of science back in the Middle Ages. Now, all science was carried out by individuals who communicated in Latin. Latin is a dead language. And even in the Middle Ages, it was a dead language. But the thing is if you could communicate in a dead language, you were assured of one thing. The language never changes. And anyone who communicates in that language knows exactly what you're saying. It doesn't matter whether they're in Italy or whether they're in Germany or whether they're in North Africa. Obviously, the world was a lot smaller then than it is now, but the same thing's true today. So originally, scientific names were essentially descriptions. You'll see the bullet point there says historical polynomial nomenclature. What that's saying is that historically scientific names started off as polynomials. A polynomial is a name with a lot of different parts. So for example, if I was to take a look at a publication from, for example, the 1500s, and some scientist was describing a species of the potato or nightshade family. Well, this plant has a description written in Latin: Solanum brasilianum, folio integro, mucornato, glabro, papas amarnacui, floribus insumitate caudis. I am not a Latin scholar, and I've probably massacred that, but it is nothing more than a Latin phrase telling me everything about this particular species of nightshade. Obviously from Brazil, obviously with certain characteristics of the leaves, that's folio. Mucronado talks about the tip of the leaf. Glabro says that the leaf surface is smooth. Pappus floribus insumati is telling me about where the flowers are. So here is a description that tells me all of the features of this plant that really establish it as being unique from all other nightshades, from all other plants. Well, polynomials were frankly very, very cumbersome because it got to the point where in order to talk about a plant, you would have to basically utter the entire description to define or identify the plant you're talking about. What a hassle that would have been. So it's much easier to basically just come up with an abbreviated version of that name. It took until the 1700s for a guy named Linnaeus. His official name, he was a Swede, was Carl Linné. And they have Latinized his name to Carolus Linnaeus, which is really, really romantic sounding, but he is the father of modern taxonomy. And what Linnaeus did was he took these long polynomials and abbreviated them into a two-part name. A two-part name is a binomial, and it's why we refer to his system as binomial nomenclature. It's nothing more than an abbreviated version of being able to specifically refer to a particular species using a much shorter, much abbreviated, and much more effective way of communicating about this particular plant or animal or species, no matter what type it is. Scientific names are two-part names owing to the work of Linnaeus back in the 18th century. Every species has a two-part name. That's binomial nomenclature. That name is commonly referred to as the scientific name or sometimes called the species name. The scientific name or the species name is always in Latin, and the two parts are, respectively, the genus name and the specific epithet. If you take a look at the name of the white oak, it's Quercus alba. The name Quercus is the genus name. It takes the part of speech of a proper noun. Proper noun is always capitalized. The second part of the name is called the specific epithet. Do not be confused. Do not call the second part of the name the species name. The species name is the entire two-part name. The second name is called the specific epithet, and technically it is an adjective. It is a descriptor. It is a descriptor that describes what's going on with the genus name. For example, the word alba in Latin means white. So that is a descriptor for this particular type of oak. Quercus alba is a white oak. One more comment about binomial nomenclature and scientific naming, and has to do with the potential for confusion that arises when we rely on common or local or colloquial names. So, for example, you'll see here that the name white oak is the common name for Quercus alba. And pretty much everybody in the United States knows what a white oak is. And I suppose pretty much everybody in Europe and in Great Britain knows what a white oak is. Problem exists is when you start going to some other countries where white oaks aren't standard. There's other possibilities for confusion as well. Right here in northwest Georgia, there is a plant that grows out in the woods called wintergreen. It's a pretty little plant that grows very, very low to the ground, and it is, as its name implies, green all year round. Well, the problem is that wintergreen is not exclusive to that particular plant. The particular plant that name wintergreen applies to is a plant with the scientific name Chimaphila maculata. I am not going to ask you to remember that name. However, that common name wintergreen also applies to at least 2 other different species. So here you got one common name being applied to at least 3 different species in one common locality. So if you were to throw around the word wintergreen, the person listening to you wouldn't quite know which of those plants you're talking about. And the same thing would be true for any other location on the face of the planet. The key to scientific communication is to make sure that you're clear and accurate in terms of what you're referring to. Not only what you're referring to, but to make sure that your listener knows what you're referring to as well. Be sure that you know the 2 parts of a binomial name. The first name is the genus name. It takes proper noun as a part of speech. The second name is the specific epithet, and it takes adjective as a part of speech. When the scientific name is written, it is either underlined as indicated by the first version of Quercus alba there, or more frequently, it is going to be italicized. And so when you're reading a scientific name in text, it is set apart from the surrounding text by virtue of the fact that it is either A, underlined, or B, italicized. And in communicating scientifically, the scientific name or the species name is the preferred method of referring to that plant or animal. All right, so at this point, we've pretty much covered systematics. We know what it's about, we know it involves classification, we know it involves taxonomy. So here's a bit of review, and then we'll be talking about phylogenetic or evolutionary trees. Systematics is placing species in appropriate groups. Those groups are called taxa. And in doing so, we establish evolutionary relationships between those taxa. We have an indication of where those taxa have come from, what their ancestors are, and what their distinguishing characteristics are. Well, one of the most important aspects of systematics, or arranging and categorizing organisms into groups, is to be able to diagrammatically represent those groups, present a visual image that will allow the viewer to understand where these taxa have come from and where they're going to. So this particular slide is going to introduce us to the topic of phylogenetic trees, sometimes called evolutionary trees, and more officially nowadays known as cladograms. Charles Darwin, in considering his results from his travels on the HMS Beagle, first drew what is widely recognized as the first evolutionary tree diagram way back in 1837, I believe. And at that point, he would have been not even 30 years old. And so there's a picture of Charles Darwin, and there's the picture of his scribbled tree in his notebook. So it didn't take long before scientists realized that if you could probably draw some kind of tree where the ends of the branches were the taxa or the groups we see today, and those branches led back to a trunk, which ostensibly would be the ancestor. And so at the bottom of the slide, you see 2 representations of evolutionary trees. The one on the right is much more traditional. You see the trunk, you see the branches. At the base of the trunk is where you would have the ancestral type, and then you have all of these derived groups coming from that. Every single branch of that tree represents an evolutionary line of evolution. On the left, you see a much more mechanistic view of the same thing, but it does exactly what the tree on the right does, but it's a lot more precise in terms of what it shows. We've got current species A, B, C, D, and E, and we can basically take a look at the shared characteristics and the derived characteristics and all of the information we have mentioned thus far in this presentation and follow those organisms back to their common ancestors and follow them back even further to even further common ancestors to a point where we have this whole evolutionarily cohesive group. So the diagram represents evolutionary relationships, the individual branches. Each line on a cladogram or on a phylogenetic tree represents a line of evolution. They are individually called clades. A clade is a line of evolution. It consists of an evolutionarily cohesive group of organisms having some common ancestor. And a taxon with a common ancestor is said to be monophyletic, i.e., one origin. So looking at this slide, we've got a picture of a typical cladogram. On the left, it shows me the current vertebrate groups, and we see the current vertebrate groups, these are animals with backbones, have some degree of relatedness. And so each line represents a line of evolution leading to, for example, the mammals, or to the bony fishes, or to the cartilaginous fishes, or to the birds, and we see how they're all arranged. And the indication is that these lines of evolution follow back to common ancestors. And so every single one of those vertebrate groups has a common ancestor. They are described as being monophyletic. So you take a look at the diagram at the top right, you will see once again a cladogram, a diagrammatic representation showing us a series of red currently existing species, and they're all highlighted in red, showing me that they represent this cohesive group that can be traced back to some common ancestor. Well, then my next bullet point shows me the term polyphyletic. Now, the word polyphyletic implies that I've got a number of groups that don't have common ancestry. They have multiple ancestry. How would that arise? Well, that's generally a consequence of some of the limits of what we know. Let me give you an example. The kingdom Protista consists of a large number of groups that, for all intents and purposes, are linked into one big kingdom because they are, number one, unicellular, number two, they are motile, and number three, they're eukaryotic. However, when we take a really, really close look at the individual groups within the kingdom Protista, and there's many of them, they do not demonstrate any degree of evolutionary cohesiveness. The kingdom Protista, in a word, is like a big, giant can full of a bunch of unrelated stuff. So I take a look at the cladogram, which is the second from the top on the right-hand side. We see 1, 2, 3 red dots indicating current species, and each of those current species has its own clade, has its own line of evolution, but we see they are not necessarily related. That would be described by the word polyphyletic. So any given decision made by any evolutionary biologist on what to include within a taxon and how to draw or represent the evolutionary history of that taxon is going to result from the interpretation of evolutionary relationships. And at this point, we understand there's a lot of information that can go into that. So how do I resolve all that if I'm an evolutionary biologist? Well, there are a number of principles that guide evolutionary biologists in making these decisions. Two of them are listed below. One is called maximum parsimony, and the other is called maximum likelihood. Maximum parsimony equals minimum complexity. There is a philosophical idea that was generated by a fellow by the name of William of Ockham way back in the 1200s. He was a monk. He developed this theorem referred to as Occam's razor. It is the idea that, now I see his quote there, entities should not be multiplied beyond necessity. It is basically the idea that the simplest explanation is the best explanation. Then there is the, I guess, flip side of the same coin, which is maximum likelihood. The fact of the matter is, if you're looking for the simplest explanation, you also want to look for the one that is most likely. And so those two principles: maximum parsimony or minimum complexity and maximum likelihood, guide the decision-making process in terms of trying to resolve these origins of these lines of evolution and where our current set of species ultimately have come from.